Live on Facebook.
Okay, so today we are having our monthly webinar for the Alliance about the yellow-legged frogs. And our presenter today is Tom Smith with the Sierra Nevada Aquatic Research Lab. And so I'm going to pass it over to him and let him start speaking. Whenever you're ready, Tom.
Thanks. I'm going to go ahead and start the screen share. Okay, great. Yes, as Sydney said, my name is Tom Smith. I am an assistant research biologist at UC Santa Barbara with the Earth Research Institute, and also with UC Santa Barbara's Sierra Nevada Aquatic Research Laboratory, which is located in Mammoth Lakes. It's one of the University of California natural reserves. There's about 40 different reserves that are managed by different UC campuses. And Snarl, as we like to call it, is one of the reserves managed by UC Santa Barbara. I've been working on this research for about 15 years, both prior to, during, and since graduate school. And so I'm going to go ahead and tell you guys today about the mountain yellow-legged frogs of the Sierra Nevada, a little bit about their natural history, but a lot about the threats that they face and some of the recovery actions that we're pursuing for them. So, and hopefully be able to answer some questions towards the end. Okay, first, before I get started, I just want to acknowledge a whole heck of a lot of people who are involved in this. Roland A. Knapp is my collaborator, co-principal investigator. He was also my boss at one time when I was a seasonal field biologist. And it's great to have had him as a mentor and sort of be able to work side by side now. Erica Hegeman and Alexa Lindauer are both project scientists that work with us. Bree Rosenblum and Sherry Briggs are both faculty in different UC campuses who have collaborated with us for a long time on this. And Max Joseph is a data scientist at CU Boulder Earth Lab, and he also was a seasonal biologist on this project way back. We also work really closely with the National Park Service, both for Yosemite and Sequoia and Kings Canyon National Parks. And we work closely with a lot of other agencies as well, including the National Forest. But the California Department of Fish and Wildlife really has management of wildlife on those forests. So we work with regions 2, 4, and 6. We work with the Lake Tahoe Basin Management Unit as well, with some projects up there. We work with really closely, and a lot of the work I talk about today would not be possible without the San Francisco Zoo and the Oakland Zoo and their staff. And we all sort of collaborate within this group loosely called the Mountain Yellow-Legged Frog Interagency Conservation Strategy Team. And this is a group of academic scientists, managers, and sort of agency scientists, as well as others, such as those from the zoo, some folks from nonprofits, where we sort of work together to align the science and the policy and pursue different priorities for mountain yellow-legged frog conservation. And lastly, of course, we're funded by the National Science Foundation, the National Park Service, California Department of Fish and Wildlife, U.S. Fish and Wildlife Service, as well as a lot of private dollars that we get through Yosemite Conservancy and the Sequoia Parks Conservancy, and some private donors that give to us directly. So, yeah, with that, I'll move forward. Just here's an outline of what we'll be talking about today. I'll give you a brief overview of the frogs, their life cycle, habitat, and some of their history. And then I'll talk at length about their declines and the causes of those declines, including introduced predators and disease, and some of the things we've been doing to recover them from both those impacts. And lastly, I'll try to wrap up with some summary and takeaway points and hopefully answer questions at the end. So the mountain yellow-legged frogs is a species complex of two species, Rana muscosa and Rana sierrae, which are the southern mountain yellow-legged frog and the Sierra Nevada yellow-legged frog. As I'll show you on a map in a moment, the Sierra Nevada yellow-legged frog occupies the northern half of the Sierra Nevada, whereas the southern, perhaps obviously, occupies the southern half, but it also, Rana muscosa lives in the transverse ranges in Southern California. And I won't be talking about those populations down there at all today. They experience a different set of conservation threats, as well as have pretty different ecology, and the restoration methods there are pretty different, and we're not involved in that. That's handled by a different set of people who we are in communication with, but they have a lot of different challenges, just sort of a different, almost a different animal, even though they're genetically the same. You can see here, actually one point before we go on, both these species are listed on the endangered species lists for both California and the United States. So they're both protected, and there are recovery plans for both of them in development. Because they're largely ecologically similar, the genetics aren't entirely worked out, and the distinction between the two frogs may change over time. The conservation strategy basically approaches them as one species, with a lot of nuance in there for different genetic clades and things like that. But here on the top, we have the adults for both frog species. As you can see, they're both pretty well camouflaged. I would say that these are very different looking individuals of the two species, but often they do not look that different, the two species. They're often indistinguishable. And for a long time, it was a single species. It was only parsed out in 2007 into separate species based on genetics and some very, some morphological features that wouldn't be noticeable to the naked eye for most of us. We have the tadpoles down below. Of course, they're aquatic. Here are eggs for both frogs. Actually, I think these are both Rana sierrae eggs. They're both sort of unusual clutches in that the one on the left is rather small, only about 20 eggs. Usually these guys lay 50 to 100 eggs. But this one on the left is more typical in being sort of underwater, tucked under a rock or something, so it's really hard to see. Versus those on the right are in grass, which is pretty unusual for the mountain yellow-legged frogs, but it is a really big clutch. And you might, if you're out hiking around to the lakes right as snow is melting, you might see some mating frogs or some eggs. Really, when the lakes are still partly frozen, the frogs are becoming active. And you might confuse them with the eggs of the Pacific tree frog, would be the other frog that you might see eggs for. But largely, you don't see the mountain yellow-legged frog legs because they do like to put them up underneath banks and rocks and things like that. This situation on the right here with grass is you would actually expect to see the Pacific tree frog eggs more in a habitat like that, in a shallow pond or something. So anyway, the tadpoles are, for the mountain yellow-legged frogs, for both species, are fairly unique in that they, from other tadpoles of other frog species, in that they overwinter for 1 or 2, maybe 3 winters. They could be 2 or 3 years old by the time they turn into frogs. And that's relatively unique. Most frogs are kind of metamorphose quickly and get out of your habitat before it dries up. But these lakes are high elevation, they're cold, they're relatively nutrient poor. And so we think that's one reason these tadpoles live a long time. These lakes also that they breed in tend to be deep enough that they don't freeze all the way during the winter. So even in a winter like this, which is a relatively low snow year, the ice on these lakes is going to be about 2 meters thick. So the lakes that these frogs are living in have to be, that the tadpoles are living in have to be deeper than that so that they don't freeze to the bottom. And these massive aggregations of these tadpoles are pretty common where there's still a healthy big population of frogs. They like to all crowd into the warmest area of the lake and get on top of each other and swim around and just hang out together during the day when it's warm. And then they swim back into the deep water at night when these surface waters cool off. Here's some adults. Again, these are in big, healthy populations. You have pretty high densities of adult frogs, and they like to crowd up and lay on top of each other and kind of form these, we call them cuddle puddles, and where all the frogs are just kind of lying on top of each other. This log in the middle, I think I tried to count it once and guessed there were about 50 frogs that you can see there. If you know which set of eyes is really a frog, which little bump is really a set of eyes. And they do like to hang out right along the water line, either on a rock or a log, so that they're always really close to the water. They might be up in these shrubby banks like you can see in the back of the central panel. These are more or less the range of water bodies that the frogs will inhabit across the Sierra Nevada. So in the upper left, we have these really high elevation lakes that have very little vegetation around them. This lake's probably up around 11,500 feet. And there's pretty much, it's a low productivity lake, it's all rock, but you'll have frogs breeding in there, tons of tadpoles living in a lake like that. And all the way down, as you sort of move across elevation, you get more vegetation in these lakes and more trees around them. And you also get into more meadowy habitat. And you can see here in the lower right panels, some meadow habitat where we have springs and streams growing in these meadows. And the frogs like those just as much as they like the big lakes. So whether it's a high unforested lake or a low elevation wet meadow, the mountain yellow-legged frogs tend to be pretty happy in there. This is the range of the frogs. Historically, they occurred more or less across the entire Sierra Nevada. If you look at these two blue blobs, that largely represents the high elevations of the Sierra Nevada mountain range. The dark blue is Ranasiri, the Sierra Nevada yellow-legged frog in the north, and the lighter blue is the southern frog, Rana moscosa, which I mentioned is split into the southern Sierra and the transverse ranges. These two black lines represent Yosemite National Park to the north and Sequoia Kings Canyon National Park to the south. And the reason I'm drawing these two areas out is just that's where a lot of the frogs remain extant today, due in part to the protections that those parks give them. But also, just based on that frogs are, that's where a lot of our research has occurred. And so I'll be referring to those areas a little more than other areas. And of course, a lot of folks are familiar with Tahoe. That's up in that sort of angular corner of California up there. And there are frogs extant up there today. And we have done a few projects with those recently as well. So, moving into talking about frog declines, this is the same graphic, but now try to imagine that most of this area does not include frogs anymore. As I mentioned, there's some up near Tahoe. Most of the big frog populations remaining are in Yosemite or in Sequoia Kings Canyon National Park, or just outside of them. There's a few sort of along the northeast side of Kings Canyon National Park. And even within those, they're greatly reduced from what they were. In total, frogs have been sort of pushed out of about 90 to 95% of the habitat that they once occupied. And that is largely due to these beasts, fish, introduced predators, largely trout, were brought into Sierra Lakes beginning when European settlers started moving into Eastern California, and includes a number of species of trout that were stocked into these lakes. Trout were fish across the board were historically excluded from these lakes by large waterfalls that they couldn't swim up. If you picture Yosemite Valley, it's the extreme case, but you have these giant, hundreds of foot waterfalls that obviously nothing can swim up. after the last glaciers receded, there was a lot of terrain like that that prevented animals from moving up those streams. But frogs are pretty good climbers and pretty strong swimmers, so they were able to get up and around and into those lakes that the glaciers left after the last recession. Initially, the fish were stocked probably just by individuals with coffee cans or whatever, but eventually it got scaled up. They were bringing them in mule trains. In the 20th century, post-war, they were bringing them in by plane as their technology was advancing, and people also had a lot more free and recreational time and money to go play in the mountains. A lot of these, so the fish stocking effort was really scaled up. And there wasn't any great science at that point indicating that there might be potential effects of introducing these species into these habitats that had where the top predator was probably the garter snake, which is a terrestrial predator even, and the top aquatic animal at the top of the food chain was probably the frog prior to fish being introduced. But as you can see in the right-hand panel, even a relatively small frog could fit relatively easily into a large fish's mouth. And if you think about tadpoles, they're pretty easily consumable by a fish. Most fish are pretty voracious predators, and being gape limited, they'll eat whatever fits in their mouth. This isn't actually a mountain yellow-legged frog, but it is a good picture juxtaposing a frog against a fish's maw. So yeah, the fish, where they were introduced, had a pretty big impact on frog populations, largely by eating the tadpoles. And this figure here indicates that. So in the top panel, we have the percentage of Sierra lakes containing frogs. And in the lower panel, we have the abundance of frogs in those lakes, expressed as a density, number of frogs per meter of shoreline. And the two bars to focus on here are the white bar and the black bar, I would say. The white bar representing lakes that fish have never been put into, that remain fishless to this day. And the black bar being where fish were stocked and remained at the time of this study, which was 2001. And you can see in both cases that the percentage of lakes that contain frogs and the abundance of frogs in those lakes is much higher where fish are absent, and that where fish have been stocked, many fewer of those lakes actually contain frogs, and the abundance of frogs is really low. They're not entirely mutually exclusive. If there are frogs in a fish-containing lake, though, they're largely pushed to the side into the fringing habitats, where the fish can't get to. And often it's just the adult frogs, and they're not breeding in that lake. Again, as I mentioned, the tadpoles need deep water lakes, and that's exactly what the fish needs. So there's a pretty clear overlap between the two habitats that the predator and the prey need. And so largely where fish and frogs are coexisting, it's just adult frogs moving into those lakes and not forming breeding populations where the fish are. All that said, it's actually pretty quote unquote easy to get rid of fish from a lake. This has been done all over the world. In a lot of the mountain areas of North America, fish were stocked into lakes, and since fish eradication programs have been started a lot on public lands, national parks, etc. It's easy if you have the manpower. You stretch a net, gill netting, you can stretch a net across the lake and collect the fish that swim into that gill net. But again, it takes a lot of effort. It's technically easy, but in terms of physical effort, it's actually pretty daunting. Here's two images, and I'll focus first on the one on the left. And this shows a little bit, these both show the success of restoring a frog population by removing fish. And so in the left-hand panel, we have across the x-axis is year, and across the y-axis are different abundances of tadpoles and adults. Densities of tadpoles and adult frogs. And these are largely for the Sierra Nevada yellow-legged frog in northern Kings Canyon and adjacent. The arrows, the vertical down-pointing arrows, indicate when fish eradication was started at these lakes. And then both all the lines indicate the densities of the frogs and the tadpoles. So in these areas, there were frogs living in adjacent ponds that were too small to sustain fish. There may not have been a lot of tadpoles there, but there were a few adults hanging on, and they were able to move back into the lakes where the fish were eradicated from. And you can see that in some cases, quite rapidly, those frog populations are growing exponentially. And for the most part, these populations stayed big for a long time. Fish eradication was quite successful in these three sites. Not only is fish eradication benefiting the frogs, it's benefiting all of the aquatic insects that live in the lake that the fish eat, which includes mayflies and beetles and caddisflies and a bunch of other things. So all those things that you would tie a fly for to go fishing are the things that the fish will eat. And if the fish is the non-native predator, eventually it eats the better part of those. So the fish were really reshaping the ecosystems in these lakes. And by eradicating them, we've restored the frogs, but also all of the other ecosystem properties that went along with that. And so that's in the short term. On the right-hand panel, we can see more long-term that some population modeling work based on population surveys throughout Yosemite shows that over the course of about 15 plus years, the average number of frogs at a site has gone up in Yosemite across the board, and that's largely due to the effect of fish eradication, allowing bigger populations to take off and grow. I think it's something about 50 lakes now in the Sierra Nevada have had the fish completely removed from them. And so you may hear some naysayers talk about, removing fish isn't fair. There's not going to be any areas left to fish. There are thousands of lakes in the Sierra Nevada. Our database alone has almost 9,000 lakes in it. Many of those have fish in them still and always will. If we removed 50 from 9,000, that's a pretty tiny fraction. So, but despite that success at removing fish and having frogs respond really positively, at the same time, around when a lot of that work was starting, scientists kept noticing that frogs were disappearing, and they were largely disappearing in places that fish hadn't been, where the frog population remained large because fish had never been introduced. And yet the populations were disappearing. Sometimes mysteriously, a survey would be done one year, and the next year it was surveyed, there would be no frogs whatsoever there. And sometimes they actually found these. This is all around in the early 2000s before I got involved in this work, but these pictures are more recent, but they were finding piles of dead frogs in some of these places. And often they wouldn't find anything. They would just, one year there'd be frogs, one year there wouldn't. If we get lucky, we actually find a pile of dead frogs, quote unquote lucky. It's not a great thing to see, but at least you know that the frogs were dying. And this was all happening at a time when people were observing amphibian declines and amphibian mortality events around the world and trying to figure out the cause of those declines and mortality events. And some of it was attributed to pesticides and some attributed to other things. But in the long run, one disease came out as the culprit, and that's this fungal pathogen, the amphibian chytrid fungus, which the long name is Batrachochytrium dendrocutitis. I will either call it just chytrid or BD from here on out. And it's responsible for a bulk of the amphibian declines that have been happening worldwide. And so you may have heard this idea that we're in the midst of a sixth mass extinction, and a lot of that is due to the declines that we've seen in amphibian species. And a lot of that is driven not just by habitat destruction or harvesting or other environmental change, but by the direct cause of this one pathogen, which is BD. And so there's here on the right is several of hundreds of papers describing the movement of this fungal pathogen through amphibian populations. So And this is all around 2000 to 2005 when people are figuring this out. So coming back to the disease, or to the distribution of the frogs, it's, I mentioned that about 90% of this original range has already been reduced by fish, and we're now seeing this disease hitting the remaining populations. And so I'm going to zoom in for a moment on Kings Canyon here. So remember this black outline, we're going to zoom in on that. And here we're just going to talk about Kings Canyon for a moment. So these green triangles represent the historical localities of mountain yellow-legged frogs in Kings Canyon, and those largely represent the higher elevation lake basins. Where they're absent are actually the really steep canyon walls or the canyon bottoms, but basically everywhere above that at the high elevations, there's lake basins, and almost all those lakes were occupied by mountain yellow-legged frogs. Beginning in around 2000, when my colleagues started working on this, a lot of those populations had already been extirpated either by the introduction of fish, some more mysteriously, and it was probably disease outbreaks that had happened earlier than that. And so we actually, over the past 20 years, I guess, have been mapping the spread of this disease throughout Kings Canyon National Park. And the white circles represent places where the disease has not occurred at that time. The red are where, in the years pictured, the disease showed up and started killing frogs. And the yellow dots are where that disease has moved through and basically driven the frog population to local extinction. There's no frogs left there, largely due just to the effects of this disease. And so you can see it's broken into chunks of years to make it a little more palatable, but you can see that those red dots sort of expand and move throughout the range of the frogs in Sequoia Kings Canyon, sort of creeping northward and into the remaining populations. You can see also that those BD negative white circles are slowly disappearing throughout each panel, and that the yellow circles where the fungus has been and frogs are now largely extinct, that area is expanding. And so as of the end of this map, which is 2017, and I'm grateful to say that for the most part, this remains the status of frog populations in Sequoia Kings Canyon National Park, is that there's this one group of BD negative sites, which is actually at the southern edge of Kings Canyon National Park, just where the two parks border each other. So that's sort of the last bastion of healthy big frog populations in that area that are completely disease-free. And I'm going to focus, before I focus on the words, let's focus on the figure to the right. This is a figure of what's actually happening during one of these disease outbreaks. So let's start in the lower panel. On the x-axis, we have days since the start of the frog population decline, which is 0, and negative days indicates some time before that population started declining, and then we move into the future. On the x-axis for this lower panel, we have the infection intensity, which is basically how sick a frog can be. It's how many fungal spores are living on the frog at a time. And you can see that as we move towards the start of the frog decline, those numbers, and this is, these are each of the averages for a different site or a different lake. And so as we move towards this 0 point, these infection intensities are increasing pretty rapidly, and the frogs are getting sicker. There's more disease on them. Moving up into the next panel, same x-axis, but here we have the percent of frogs infected in a population. So this is, the word for this is prevalence, which is the number of individuals that have a disease in a population, or the percentage. And we can see that it's going from 0 prior, it's ramping up really quickly to almost all those frogs being infected in a lot of these populations. And then the data actually ends largely because of the result in the top panel, which is the number of frogs. And so here we have um, you know, frog populations numbering in the hundreds to thousands up here. And they stay stable for a little while. And actually, as the disease is getting worse in the population, you don't see a lot of mortality. And then all of a sudden, they all go off a cliff and basically go to zero. And the disease, because so many frogs are so sick, as we see in the bottom panel, and because so many of them are sick, as we see in the middle panel, more or less, those populations go to extinction. And so this disease consistently, when it emerges in a frog population, is driving them extinct. That's something we call epizootic disease dynamics, where it's sort of the animal equivalent of the word epidemic, which we use for humans, and we should all be pretty familiar with this winter and spring, unfortunately. So when we have epizootics happening in these frog populations, we're seeing mass mortality leading to local extinction. However, there's a very interesting fact that is actually good. Not all frog populations are equally susceptible. In many, the infection intensities don't get as high, and the percentage of frogs doesn't get as high. And so you actually have some frogs surviving, and the populations aren't getting driven to local extinction. There's still high mortality in those populations, but because it's lower than it is in the other situation, these populations actually persist throughout time. And so that's actually given us one of the tools that we use for frog restoration, because we now have these frogs who seem to be resistant to the disease in some regard. There is actually a geographic component to this, wherein, as I showed you for Sequoia Kings Canyon National Park, that almost wave of disease moving through the park, those have all been epizootics. And we're only now getting an idea that there might be some enzootic populations throughout that area. Meanwhile, across the same period, Yosemite National Park has appeared to primarily be inhabited by enzootic populations, where most of the populations in Yosemite, they have the disease. Some of the frogs are dying, but some of them surviving, and they continue to breed. And those tadpoles are successfully turning into adults. So over a long period, we haven't seen a lot of change in those populations in Yosemite. What we're hoping for is to find more of those populations throughout the Sierra Nevada, because as I mentioned, they're pretty critical to our ability to try to restore frogs throughout the range. If frogs are susceptible to the disease, you can imagine that anywhere we move them, they're just going to die there as well. So a lot of our ongoing research is into the cause of this difference in susceptibility between different frog populations. Is it a difference in the frogs? Are they genetically different? Or are their immune systems operating differently for some reason? Is it the fungus? Are there genetic differences in the fungus? There probably are, and there's some evidence that suggests there are, but by and large, the fungus appears pretty similar across the range of the mountain yellow-legged frogs. Or is it just time? Is it natural selection, selecting the animals who are going to survive something needs time to occur, right? It doesn't happen quickly. That's one of the challenges to documenting it and showing that it's a real phenomenon. And so it's possible that these outbreaks happened in Yosemite decades before they started in Sequoia Kings Canyon National Park, and that's why we see enzootic populations in Yosemite versus they're still the disease is moving through Kings Canyon for the first time. And so it's causing epizootics, and that bottleneck is still selecting the frogs that can survive it. So that's all ongoing research. To do that work, we do a lot of frog surveys. You can see this gentleman here walking around looking for a frog that he's going to catch in that net. He's got two things carrying with him around his neck. In this pouch is a pit tag reader. So in a lot of the populations we study, each frog has an individual tag under its skin. If you have a pet and you have it tagged, so you can scan it and this little number comes up, it's pretty much the same technology as your pet might have. Once we find a frog, you can see Alexa here is taking a skin swab, and this is actually pretty similar to the swabs that people are getting done for COVID-19 right now. It is a little bit different. So we're just trying to take some of the frog's skin. The fungus lives in the frog's skin, so we're trying to take some of that frog skin, and then the DNA of the fungus and the frog are in that skin, are on that swab, and we can actually do a lot of genetic-based work based on the fungal cells and the frog skin cells that are in that swab. So there's a lot of research and actions that we do to try to recover some of these frog populations. So where we have persistent frog populations, we're trying to do translocations, and that's what this helicopter represents, is that we're often collecting frogs at a site, and then the Park Service or whoever is helping us actually fly those frogs to a different lake where they can be released. And this often happens after fish have been eradicated. And so once the fish are removed from a site, we can actually then release frogs into it. And if we use disease-resistant frogs, all the better, because they'll survive on their own. This lower left panel here, you can see these little froggy faces all pressed up against the side of this plastic bin. This is actually something we're still studying, whether a sort of antifungal treatment can save these frogs. There's a lot of lab experiments that show that you can actually soak these frogs in an antifungal solution. It kills the fungus, or some of the fungus on the frog, and that actually can buy them time for their immune system to work. You know, in those pictures I showed where the frogs were getting sick really fast in that graph, that rate of increase is actually exceeding the rate of increase that their immune system can respond to. And so we're actually trying to buy these frogs time. We're not trying to cure them entirely, but basically try to buy them time where their own immune response can react. So we treat them in this antifungal, and we're trying to figure out whether we can do that in the field. And it's very difficult because it requires a lot of manpower and it's stressful for the frogs. They do this pretty regular. A lot of the frogs are raised in the zoo, which I'll talk about in a moment. And they do this regularly in the zoo and in a lot of laboratory populations so that their animals are living disease-free, which makes them a lot easier to take care of. These tadpoles up here were brought from the field, probably in a helicopter, and then driven to Oakland or San Francisco Zoo, and they get raised there into big fat frogs. The nice thing about the zoo is they can turn tadpoles into really fat adult frogs pretty quickly, and also with a really high success. So the survival rate of those tadpoles is much higher, and they can be released into the field again to sites where fish have been eradicated for the most part. And then down here in the lower right, we see this frog taking its freedom from this little plastic cup and swimming off into a lake, probably for that frog for the first time. And it's really, really nice to see them getting back into the wild. So some of the things we've learned, and I'll walk you through this figure in a second to illustrate this point, is that these frogs, as I mentioned, the tadpoles live underwater for a couple winters. And these are cold environments, so they grow pretty slowly. A frog might be 5 or 6 years old until it reproduces. So time, patience, and persistence have been something we've learned in these efforts to translocate and reintroduce frogs, is that it just takes a lot of waiting and a lot of watching. So this figure describes 2 frog translocation projects we did in Yosemite, one starting in 2005 and the other starting in 2008, where 40 and 36 frogs were introduced into each lake. And you can see that we went back several times. Each dot represents one survey. So many, many times during all these years, we went back to these populations and counted the frogs. And the other dashed vertical lines represent subsequent translocations of frogs. So we were trying to give them a little, sort of subsidize that population a little bit. And we did that in 2013 in the upper lake, and in the lower lake, we did it in 2015 and 17 as well. You can see for the upper lake is especially clear. There's this bump in new frogs. The frog abundance starts to go up in about 2013 and 14. So that's what, 6 or 7 years after we started this project. And then it sort of stayed stable for another year. It took a huge jump in 2016. Since 2017, 18, and 19, it's been stable. And we're wondering what it's going to be like this year. We don't really know. So like these two sort of bumps in recruitment, we say, that's the word for new frogs entering the population, were a little bit unpredictable. And it took a really long time from when that project started to when that population became a really big, healthy, thriving population. You can see for this lower population, we made several translocations and introductions of frogs. And that population has just never really gotten big like we hoped it would. And we're not quite sure what the difference here is. So time, patience, and persistence pay off in the top example, and we're still trying to figure out, through those three, time, patience, and persistence, what's going to happen with this lower one. But it's been about 10 plus years to really see this top population take off and become a big, stable, thriving frog population. A second really important lesson that we've learned is that collaboration and communication are absolutely key between the science, which is largely us, and the policymakers, which is the Park Service, Forest Service, CDFW, and US Fish and Wildlife Service. We all have to talk a lot to make sure that we're working on the same priorities and getting all the hoops and things together, jumping through all the right hoops. And we've actually got a pretty great group of folks who work really well together, and that has helped a lot. So yeah, just to review a little bit, I talked a little bit about the life cycle of these frogs with long-lived tadpoles, 2 different species living in the north and south. They used to be extremely abundant and fairly ubiquitous, but have largely declined from up to 90% of their range due to introduced predators, which is a reversible impact, and disease, which so far we're kind of still in the research phase of figuring out how to reverse the impacts of this disease. So why do these frog declines matter? Well, for one, these frogs are part of a food web that has been around for thousands of years, since the last glaciation, which was somewhere in the range of 15,000 years ago. And frogs here are this sort of lavender circle, and you can see that they have many connections to different insects, as well as garter snakes. And we've done a project looking at garter snake predation on these frogs. As you can see here on the right, the snakes are the natural predator of these frogs and have been for, as long as they've both been living in these habitats. And so, the frogs impact this entire ecosystem, much as the fish did when they were introduced. Removing the frogs can have dramatic impacts on some of these other species, especially those who depend on them almost exclusively, which are the garter snakes. And lastly, I'll share this humorous approach to why this question of why do frog declines matter. This was shared by one of my colleagues. We can actually learn, when we think about wildlife disease, we can learn more about human disease. And right now we're living in a time where that actually matters. One, the disease that we're dealing with largely probably originated in wildlife, as have many others: Lyme disease, HIV, and we could go on with that list of diseases that originate in wildlife. BD, the amphibian chytrid fungus, is pretty unlikely, if not impossible, to cross over into humans, but we can still learn lessons from that. These frogs are not keeping distance. They are all on top of each other. And if that were an infected population, if one of those frogs were infected, those frogs would all be getting infected quite well. We don't know how to incorporate, we know that social distancing now works for humans, right? At least for this disease. It would probably work for these frogs as well, but we haven't figured out a way to logistically force these frogs to not contact each other. And so, there's lessons we can take back and forth. from what we've learned from human diseases and what we've learned from wildlife diseases. And although this is maybe a humorous and slightly irreverent approach to it, I think it nails that point home. So yeah, that's what I'm going to leave us on. And I'm happy to take any questions that you guys have. Thanks.
Okay, does anyone have questions? Okay, I'm guessing that no one has questions. So thank you for presenting today and taking the time.
Sure, yeah. If you go to the URL for our website, my contact info is there. More descriptions of oh, actually, let me stop the share. Maybe people will get friendly when they can see each other. You can go to our website there, and my contact info is there. And Sydney or anyone else from Sierra Nevada Alliance can probably hook you up with my contact info.
And when I do a follow-up email, I'll include your email and with your email address for everyone to be able to contact you if needed.
Yeah, that sounds great.
Okay, awesome. Thank you.
Okay, thank you, everyone. At least thanks for letting me see your names in your little black squares. And I hope you guys all have a good spring and summer season, whatever you're doing around the Sierra Nevada. We definitely appreciate your efforts, and we know that Sierra Nevada Alliance is a good part of our community. So thanks.
Thank you. Have a great day, guys.
All right, thanks, everyone.
Bye.